to the third and final installment of online lecture 3. In this lecture, we will discuss the biological catalysts known as enzymes in more detail. Specifically, we're going to focus on the protein enzymes as part of our unit on proteins. You should recall from Chapter 2 that enzymes speed up the rate of chemical reactions so that the reactions occur at a rate that is biologically relevant. Enzymes do this by lowering the activation energy of a reaction. This action of enzymes is true both for endergonic and exergonic reactions. This slide shows the effect of an enzyme on an exergonic reaction and illustrates how enzymes change activation energy that is required for a reaction to proceed, but do not change the overall delta G. So how are enzymes able to do this? Before we look at the specific mechanisms by which enzymes lower activation energy, it's important to understand the basic process involved in an enzymatic reaction. During an enzymatic reaction, the substrate binds, forming an enzyme-substrate complex. The enzyme then catalyzes the reaction, resulting in the formation of a product. The enzyme, which is unchanged, releases the product and can then catalyze other reactions. The substrate can't bind just anywhere along an enzyme. It binds to a very specific region with a specific shape, and this region is called the active site. Active sites range from approximately 6 to 12 amino acids in length. You might then be wondering what the rest of the enzyme is for. It provides the scaffolding, that is, it holds up the active site, or sometimes it can change shape, undergoing what we call a conformational change to bring two substrates together in what's called an induced fit. Now we're ready to talk about how enzymes lower activation energy. Once the substrate binds to an enzyme, there are a variety of methods in which enzymes lower the activation energy. One is to simply orient the substrates or put them together in such a way to facilitate a specific reaction. Sometimes enzymes just bend the substrate a bit to induce a strain. This is enough to weaken a bond so that a reaction will occur. Enzymes can also impart a charge to a substrate, which can facilitate certain types of chemical reactions. Enzymes do speed up chemical reactions, but the rate at which a reaction proceeds, even with an enzyme present, is affected by environmental conditions. These effects are a direct result of how the environment affects the enzyme. For example, because an enzyme must bind a substrate, the rate of a reaction will tend to increase with increasing substrate concentration, as shown here on this graph, until a maximum rate is reached. At this maximum rate, or what we would call saturation, all enzymes are very busy, and the rate can't increase anymore. In fact, it's limited by the time it takes for an enzyme to bind the substrate and then form the product. So adding substrate won't increase this reaction at all. Enzymes also respond to temperature, and often in the manner that's shown here on this graph. There is often an optimum temperature at which the maximum enzymatic activity occurs and the fastest rate. And then on either side of that, the rate of the reaction decreases, but for different reasons. Let's look at this graph here. We see that temperature is increasing along the x-axis, and that with increasing temperature, the rate of the reaction decreases. Can you think of any reason why? I want you to pause for just a second and see what you can come up with. Okay, so if you're thinking it has to do something with denaturation and something we discussed in the last section of Lecture 3, you're on the right track. But what's happening on the other side here, where the reaction rate slows down as temperatures get colder? This is basically a reflection of what's called collision theory. And collision theory basically just says that as you heat up a solution, that the molecules will move faster and tend to collide. So if you cool it down, the molecules move more slowly and are less likely to collide, which in the case of an enzymatic reaction can really slow things down. This graph shows the effect of pH on enzymatic reactions, and you'll notice that this slide looks very similar to the one we looked at before with temperature, and that generally speaking there is a maximum rate or an optimum pH where the reaction rate is the fastest, and then either at a lower or a higher pH the rate of the reaction slows down. The reason for this is that pH can have a profound effect on the structure of an enzyme, sometimes even denaturing it. And most enzymes have a precise pH at which they are most functional. It usually has something to do with where the enzyme is located in the body and the pH of that area. For example, an enzyme with an optimum pH around 2 is likely to be a digestive enzyme that would be found in the stomach. The stomach has a very low and acidic pH. An enzyme that has an optimum pH around 9.5, on the other hand, may be found in the intestine, which tends to have a basic pH. So it would be very inefficient to have enzymes active all the time in the body, particularly when they're not needed. There are actually a variety of ways in the body in which enzymes are actually controlled. And because of their roles in diseases, enzymes are often the targets of drugs or even of chemical weapons. This slide is showing an example of a type of inhibition. This is called an irreversible inhibition. The enzyme that's being targeted is called acetylcholinesterase. It is very important in nerve transmission. When this acetylcholinesterase is bound by a compound called DIPF, it actually binds irreversibly to this enzyme and inactivates it. This basically prevents the acetylcholinesterase from degrading a neurotransmitter, which ends up making the neurons fire constantly. Unfortunately, this has been capitalized by in chemical weapons, and DIPF is actually a component of a very potent nerve gas. On a more positive note, irreversible inhibition is the way that penicillin works to inactivate a bacterial enzyme. So we're going to look at some examples now of competitive and non-competitive enzymatic inhibition. So competitive inhibition occurs when there's another compound that can bind to the active site of an enzyme, and in so doing actually blocks the ability of the substrate to bind to the enzyme. Competitive inhibition can actually be overcome by high substrate concentration. Non-competitive inhibition actually works through a different mechanism. In this case, the non-competitive inhibitor actually binds to the enzyme, causes its shape to change just slightly. The substrate can still bind to the active site, but this slight change in the shape of the enzyme has actually rendered it ineffective, so that even though the substrate binds, no product is actually formed. And this is why it's called inhibition. So one example of this is actually lithium. Lithium actually prevents the metabolism of polyphosphoinositide in the brain, and that's why it's used to combat bipolar disorder. Enzymatic reactions are also controlled by a process called feedback inhibition. And this is where the accumulation of a product that is sometimes several steps away in a metabolic pathway actually inhibits an earlier step in the process, specifically by acting on the enzyme that's involved in that process. So for example, an accumulation of isoleucine can actually cause this early step where threonine is used to form alpha-ketobutyrate and inhibit this reaction. So this is one way of making sure that once there's enough isoleucine that's being made, that its synthesis is discontinued, and it's discontinued at an early stage so that there isn't a lot of energy wasted in the synthesis of isoleucine that's not needed. Finally, enzymes can also be regulated by a process called allostery. And allostery is just the changing of the shape. In the case of allostery, the enzyme is actually present in the body typically in its inactive form. What can happen then is that it can be activated sometimes by the addition of a phosphate group. The proteins involved in this are called protein kinases. The addition of the phosphate group causes a change in the shape of an enzyme and opens up the active site. The enzyme then is able to catalyze the reaction and produce a product. Sometimes enzymes are activated by compounds other than phosphates, and they are called activators. The activators then work in much the same way as the phosphate does to change the shape of the enzyme, allow it to bind the substrate, and to create a product. So this concludes online lecture 3, part 3.